Hello everyone and welcome to our NGS-CN Summer School online collection on spatial transcriptomics. Before we jump to our next speaker, let's take a look once more who we are. So it's a next-generation sequencing competence network that consists of 4 production sites, as you see on the map. So in 2019, DFG has funded centralized sequencing applications. There were quite a lot of calls, and within each call there were a lot of projects. And from 2024, DFG has changed the strategy towards individual sequencing applications. That means that now there is no special deadline for a centralized call, and you can co-apply at the DFG together with the chosen genomic facility. If you wish, here is a QR code, contact us, and then we could take a look and maybe sequence together. If you want to know something more about our news and upcoming events, then you also can take a look at the QR code, scan it, and get into our mailing list. Let's grow community together. And with that to be said, let's welcome our next speaker.
Thank you. I'm Fabian. A couple of months ago, Juli invited me, asked me whether I would give a lecture on introduction to spatial analysis, and I said yes, because I had it good. I did it once in my life, and then I started to prepare the talk, and I got a bit nervous because there's so much going on. Disclaimer: this is going to be biased. This is about the stuff that I know. I'm working in Mary Oak's lab at the MPI CBG here in Dresden. Before I worked here at the Genome Center, and I counted I touched something like 5 spatial transcriptomics datasets in my life. So it's not a hell lot, but it's some. And yeah, regarding biases, all of them, everything I worked on has been 10x. So I think you will hear a bit of 10x from me. This is basically because 10x is present here. Doesn't mean that I think that this is the best. And then in terms of, I will talk about a few analysis tools, and I use coding to do that. And I think there's 2 main programming languages, Python and R. And I'm a Python person by heart. And we should reflect on our emotional biases, I think. And if there's more Python tools, that's the reason. All right, spatial transcriptomics. I have a slide on why we might want to use spatial transcriptomics instead of single cell or spatial. I think I don't have to go through that. I think Juli did a wonderful job in explaining that. Maybe I want to point out some questions that we can specifically ask to spatial sequencing data. Those questions might be, of course, where are specific cell types located in a tissue, right? Spatial location, that's what it's all about. But also, what is the spatial arrangement of gene expression within a tissue, right? It's not necessarily directly related to cell types. How do different regions in a tissue differ transcriptionally? I mean, this is a similar thing as before, but maybe you can actually define different regions by morphology. There might be a tumor surrounding region, something like that, and you want to understand what are the differences. You may also use that to how cells communicate. There are like statistical inference frameworks and single cell data to do that, and you can extend that to spatial. And also, I think you can learn something about cell niches, right? So what is the microenvironment of specific cells? And my idea is kind of for today to introduce you to some computational methods that have been developed specifically for spatial sequencing, or also like I would say I'll start with how you can basically directly apply methods that you already heard about from Katrin yesterday on single cell RNA sequencing. Okay, I think it's pretty clear that there are like 2 main technologies in the field. We have heard that from Juli. We also heard that from 10x. There's this sequencing-based methods, which usually give you like a full transcriptome, but a bit less resolution. And there are these imaging-based methods, like Xenium or Merfish, for instance, which have even subcellular resolution. How does this spatial data actually look like, and more from an analysis perspective, like conceptually? How does this data look like? We want to analyze this data, so how does this data look like? Maybe to remind ourselves, and here I take a figure created by Katrin to remind ourselves, like, how does single cell RNA sequencing data look like? I think that's basically a big table of you have cells here denoted by barcodes, you have genes, and you note down their expression, right? And that gives you a high-dimensional state space, and you can analyze this, and Katrin introduced you to that yesterday. Maybe you can extend this a little bit. That's low resolution. Sorry for that. the idea here being that maybe you can extend that a little bit in terms of it's not only the gene expression profiles, but there's another table that annotates the genes. There's some metadata on the genes. There's like ensemble IDs, whether they are mitochondrial, whether they are ribosomal. There's some metadata on the cells, like from which sample are they from which time points, things like that. But it's basically all big bunch of data tables. And now if we go into spatial data, what additional layers of information do we have other than tables? And I think obviously, right, we have images and we somehow have to store that spatial information. And this spatial information is quite diverse in terms of technologies. Here I'm actually taking, and I want to give a shout out to Tim Treis here from SCVerse, one of the developers of the spatial data packages. And I recently attended the workshop with him, and I'm using some of his slides. And so this slide is from him, and some more slides follow. And what you can see here is this kind of like this increase, which I think we all know about in terms of spatial transcriptomics paper, spatial transcriptomics technology. And here are just a few names of spatial technologies listed. This is not just transcriptomics, also some proteomics, but it's super diverse. And in red, there are the two 10x technologies that we just heard about, Visium and Xenium. And each of these technologies, they come either many come from different companies and they produce their own data formats. And even just within the 10x ecosystem, if you just look at the Visium and the Xenium data, they come up with new versions as they adapt things. And there's always like new file formats that you have to work with. So one of the issues that you then have there is that 10x just introduced, there's things like the Xenium Explorer that you can use to analyze Xenium data. There's things like the Loop browser to analyze Visium data. But usually you cannot go from one technology to another data. And now maybe you use VisGen, Murfish at some point, but you cannot analyze this in the 10x data. So how can we overcome that? And there's like I would say in the scientific communities, there's an idea to generate some standards. And there's, for instance, this paper from 2022, which points out that standardization is a key requirement also for driving computational innovations, because you want to be able to easily access your data. Or also, if you want to compare your data, you create some data and you have some reference data set, but it was not created with 10x, but with Murfish, and you want to compare your data to. It's important to have easy accessibility, right? So, okay, before now going into like this, I think has two components. One is the technical component, like what are the file formats? One is more a conceptual component. How do we think about spatial data? And I think one thing that I really like here is this concept that comes from the spatial data package from the SCVerse to categorize spatial data in these categories. So one of the categories is like tables. This is what we know from the single cell RNA sequencing world, right? You have a cell and in a table, you write down the gene expression. In another table, you write down the metadata and so on. I think images, that's pretty obvious in the spatial world. We are all used to images. They are pixel-based. And basically here, every pixel encodes an intensity. Similar to that, you can have labels. The main difference, so this is also pixel-based. The main difference is that the intensities here now are not continuous values, but they either denote a segmentation mask or they could denote something like cell identities. Sometimes it might be more useful to store spatial data in form of points. We saw these beautiful Xenium images where every transcript is a single point. So that's not pixel-based anymore, but for each of these transcripts, you have the X and Y coordinate, maybe X, Y, and Z even. And shapes are useful, right? That is useful if we go into cell segmentation. If we want to outline a cell, we can just store that as a shape. And all these things, of course, can be connected. For instance, these shapes can be a cell. And then for this particular cell, you want to annotate that cell with a table denoting, for instance, you can, for instance, count the number of transcripts within this cell and then derive a count matrix. So this is, I think, how the data looks like. I think in my understanding, everything that you kind of can think of in spatial, you can put in this framework. And yeah, I think that's super helpful then in terms of when you start trying to analyze it. I think it also becomes clear that these data are way more complex than single cell RNA sequencing data, which are complex already. To put that in action, maybe take a moment to think about which elements are used. So here I'm showing a snapshot of a Visium data set. You can basically see the H&E staining in the back. And here are these Visium dots that were just introduced by 10x, and it's color-coded by the expression of the gene. So in which kind of spatial elements would you need to store these? We can ask the same question about Xenium dataset. So here you see this kind of the segmented cells. There's an image of DAPI in the back, I think, and you can also see transcripts. So which spatial elements would you use here? The answer was all, and I think that was perfectly right. That you can use everything here, right? You can store the images of the DAPI staining, for instance, as images. The transcripts themselves in point clouds. Segmentation masks you can store as labels and also as shapes, and then you use the tables to annotate, for instance, cells. And I basically wanted to set this stage more in terms of like how to conceptually think about these data. There's of course also then like file formats, like images can be stored as a PNG, as a TIFF, and so on. And these companies have proprietary image formats as well. Or like to use different mixtures. So we saw the complexities of the output of Xenium, for instance, all kinds of image files. And I think one of these ideas of several of these community projects that exist, like spatial data, and in the Python world and Voltron, maybe as an example in the R world, is to actually come up with readers that read all this kind of information into one standardized package that you then can provide to the community. All right. This about like how I think spatial data looks like. Now, let's dive into how to analyze these data. I think before you actually start your experiment, I think one thing where kind of data analysis can be useful when you have a single-cell RNA sequencing data set is to design probe sets. We heard before that Xenium, like image-based methods, are probe-based. So how do you come up with an optimal probe set? I won't go into that, but I just want to mention there's a nice paper that actually comes with a nice, like some nice questions if you visit the GitHub site for how to do that. And there's some software that kind of allows you from single-cell RNA sequencing to kind of come up with an optimal probe set. Once you have that and you created your data, how do you start with pre-processing and quality control? I won't go through the very initial. The initial processing is already done. We are in this world of where we now have like images, we have point clouds, we have shapes. One of the first steps that you need to do, and we heard about that before, is for imaging-based methods. If you want to analyze single cells, you need to segment the cells. This is needed to construct a cell-by-gene count matrix. Typically, this was done with nuclear segmentation. Now, more and more cell boundary staining kits are out there. I've never worked with that before so far, but I think that's super useful. Or you can also use the transcriptome for segmenting cells. And I'll touch that later if time permits. But that would be the first step. And once you have that, I'm just showing this slide here with the Visium and Xenium and Visium HD, like different technologies. One thing that all these have in common now is that you have these tables. You have this cell by gene count, cell by gene or spot by gene count matrices. And they are, of course, very similar data structures like as you use for single-cell RNA sequencing. And you can basically apply the same kinds of ideas like Katrin introduced yesterday. Like you start with quality control on your spot or cell by gene count matrix, and you, for instance, filter on, I only want to take cells or spots which have enough reads, which have enough detected genes, which have a low percentage of mitochondrial RNA. And maybe on top of that, you can think of things like cell area. I don't want to use cells that are too small or too big that might be segmentation artifacts. You go through normalization. Then you basically borrow methods from single-cell RNA sequencing again. There's like log normalization or Pearson residuals that would be SC transform in Surat. But there's also some specialized methods for spatial data. Here I'm just showing one example called STSME, which is part of the ST-Learn package. I'm always giving the links down here. Yeah, maybe one thing that I did mention, and basically my idea is I want to give you some concepts of what you can do. There are links to the methods, but I won't have time to explain the methods in detail. Basically, what the authors here propose is that they have a special method for normalizing spatial data. And here you can see a bit the difference. This is standard normalization. This is their normalization. And I think you see a bit that this looks a bit more homogeneous, a bit less noisy. How much this matters in practice, I'm not sure. There's no benchmarking out there. So far, what people use a lot is just like standard single-cell RNA sequencing methods. You can denoise things. In the interest of time, I'll skip that. One thing that I wanted to mention, just to put it as a term, because when I came from the sequencing world, just the term was new to me, is called image registration. What that means is basically, if you have two images that show the same thing, you want to put them on top of each other, you want to align them. And that process is called image registration. And that's a world in itself, if you want to do that, and how you do that best. But there are tools out there. One of them is inside the spatial data world. You can use Napari. Here you align a Visium H&E to a Xenium image, for instance, based on landmarks that you manually select. You can do that also, for instance, in the Loop browser of 10x, or I think also in the Xenium browser, you can do that. So image registration, this is basically, I think the most of the one important thing is if you have an H&E image that you take under a microscope and under one microscope, and you have, for instance, your sequencing data somewhere else, and you might want to align them, for instance, to then be able to annotate regions of interest, which, for instance, could be done by the local pathologists in your hospitals that you align this region of interest, which might be, for instance, a tumor area, and then you can compare that with your Xenium results in terms of what's the cell type composition within this region compared to without. I think that's what I want to say about the initial steps, I would say, in terms of data pre-processing. Now I want to dive a bit more into downstream analysis. And one thing that you obviously immediately can do with spatial analysis is you can plot genes in space. Here I'm just showing you two examples, two genes, and you see they nicely localized in certain areas of the brain here. If you want to plot genes, there's quite some software available. We already heard about some proprietary software by 10x, like Visium and Xenium. Really nice if you use their systems. If you go, there's one by Murphish as well for their software. Really nice if you use their system. This software is dedicated for the company's data in a way. There's some open source approaches. Napari, some of you which work with image analysis already might know Napari. This is how a Napari looks like, right? And then you can, for instance, select genes, look at them here. Vitesse is an interactive explorer for spatial transcriptomics data. And I think we will hear about that and then its connection to Omero tomorrow in a talk. Okay, and I think that having these interactive tools is super helpful for you want to be quick looking at your data, you want to select the gene, look at things. But the question then is, which genes are interesting? If you have a Xenium panel with 100 genes, you might still be able to go through all the 100 genes. But if you have 500, I don't know. If you have 20,000, you certainly don't want to do that. So which genes are interesting? And I think here what we're looking for is spatially varying genes. And what do I mean by that? So going visually here, to make a discrimination between a highly variable gene and a spatially variable gene. A highly variable gene like this gene would be a gene that varies a lot from spot to spot, but doesn't show a spatial pattern. This is just noise. We're not really interested in that. What we're more interested in is a gene that shows a particular pattern like that. This is what I would call a spatially variable gene. And how can we get them? We can make use of methods that have been long developed, I think, in the context of geography. When you study maps, a guy called Moran introduced a quantity called Moran's I, and you can see here a few properties of that. Like if you have a checkerboard pattern, you would get a perfect -1. If you have something that is pretty homogeneous and noisy, you're close to 0. If you have something that is more patterned like that, you get higher values, right? And that way you can compute this value for each of your genes, and then you can look for the things that are not 0, and those will show a spatial pattern. And that's nice. That allows you to select some genes. And I think a lot of software does that automatically for you, allows you to do that. Now, one issue with that is that this does not group genes by their expression pattern. And it might still give you a couple of hundred genes that are spatially, that show a spatial pattern, and that's interesting. But if you go through them one by one, it's like, there's like this gene is expressed on the left, this gene is expressed on the right, this is on the top. So you see all kinds of patterns and it's jumping around. So how to overcome that? There's dedicated tools. One of the tools is called Spatial DE, so Spatial Differential Expression, dedicated tool developed by the Teichmann lab to identify spatially variable genes. And here it shows you some examples of some genes that show spatial patterns. And I think it's clear that they show some interesting spatial patterns. Some things here expressed inside versus outside. I think that's some brain tissue. And what that does on on top is it allows you to identify patterns and to group genes into patterns. So, for instance, I think those two genes that we see here, right, which are expressed inside, they would be part of this pattern called here pattern 2, which is comprised of 6 genes. Yeah, spatial DE allows you to identify spatially variable genes and to group them into patterns. That's it about now visualizing genes in space. What else can we do? Of course, we can do, we have a count matrix and we know all these tools from single-cell RNA sequencing, and we can do dimensionality reduction and clustering to learn something about the cell types that we have in our system. So we apply the typical single-cell RNA sequencing workflow. We have the spot or cell by gene count matrix. We go through principal component analysis and UMAP and clustering, and we might arrive at something like that. So this is a UMAP now where every dot, this is actually created on a Xenium image, but could look similar for a Visium image. So every dot here would be either cell or a bin. And if they're close together, they're transcriptionally similar. And what I did here is I clustered them in some cell types. Now, if I would do the same thing on a single cell RNA sequencing datasets, the clusters that I see here would actually be super distinct because they are very different. There's immune cells and there's skin cells, for instance, in this dataset. And here, what I see is all these kind of mixture cell types. And now, if you think about like for Visium or also for Xenium assays, why would you expect maybe to see this kind of mixture cell types? Right, if you're in a Xenium image-based assay, if you don't get segmentation right, cells are assigned, transcripts are assigned to the wrong cell, and then things look like a mixture cell type. Or in Visium, in the grid-based assays, you can have a cell which spans multiple of these spots, or you have a mixture of cells on a particular spot, and then you get this kind of mixture cell types. You can kind of overcome that by revisiting cell segmentation in part. So here I'm also just like, I wanted to show an example where you can see these kind of mixture cell types. So here I'm denoting two molecules. CD8 marks T cells, EDNRB marks skin cells, and these are very distinct cell types. And you wouldn't expect them to be expressed in the same cell, but even within nuclei, you can kind of see here you see this segmented nuclei, and you see like there's both colors inside. And actually then if you make use of the full 3D information and kind of would rotate that, you would realize that there's actually a T cell nucleus beneath a skin cell nucleus, and this is actually 2 cells. So how to overcome that? Better segmentation. I think cell boundary kits are super useful if you have them, or you can use the transcriptome itself for segmentation. That was also mentioned in the earlier talk. What does that mean? So there's basically 2 tools, BASO and ComSeg. And what does that mean using the transcriptome for segmentation? Well, you basically make use of these transcripts that you see here and kind of make use, then you kind of learn, okay, typically these transcripts, the green ones and the pink ones, they are separated. So they should be separated here as well. And these methods then learn from this high-dimensional point cloud where the cells are. Right. And if you then look at the impact of cell segmentation, this is how the UMAP looked before. Here it's now color-coded by these 2 genes, which should be mutually exclusive because one is skin, one is T cell. This is like a DAPI-based segmentation. And if you do the segmentation then based on the transcriptome using BASO, you can see that the CD8 T cells end up here, the skin cells end up here, and they are really nicely separated. So segmentation is an issue. Now, that was basically the application of what we already know from the single-cell RNA sequencing world. Now, how can we actually make use of the spatial information, right? We have spatial data. How can we make use of that? And I think there's like many ideas and tools developed at the moment, and I want to showcase some of them. One of them I would call spatially aware clustering, where the idea is that you group individual spots or cells into clusters. This is also what we do in single-cell RNA-seq analysis, but you use both informations, the transcriptional information and also the spatial information. So you add spatial similarity to that. One example that will actually also be used in the hands-on workshop is Banksy. And there, the idea here, I sketched as an image, I won't go into the details of the method, is if you now want to understand something about this cell and what cluster it belongs to, you take the information coming from this cell directly, so the transcriptome of this cell, But on top of that, you also look into the neighborhood. So you define these cells as the neighbors, and you take their transcriptome into account. And on top of that, another layer that they add is from that, they also compute a gradient of expression, right? You also take into account whether there's like more gene expression here compared to there. And that's a third layer. So adding all these layers, now your clustering algorithm becomes aware not only of the cells themselves, but also about the microenvironment. And how does that look in practice? Here, I show you a slide from the Banksy paper. So I think that Banksy looks really good. You have to kind of take care that it's from the Banksy paper. This is a Visium HD slide, and it has been annotated manually, I think, based on the H&E staining. And these are clusterings that were generated based on the transcriptome. And the non-spatial transcriptome, this is what you kind of would do when you treat all these dots in space as if they would be individual cells, as if they would, you don't care, you forget about the space, and then you kind of group them based on transcriptional similarity. And you would say like, okay, these look transcriptionally similar. And you can see that this works quite well. It closely resembles what has been assigned as a ground truth. But you see this kind of noise in terms of space, right? Which comes due to the noise that we have in the transcriptomic profiles. Now, if you go into this Banksy approach, which takes about the spatial information into account as well, boundaries become much clearer, and the picture way more looks like what you would expect from the H&E staining. So this is spatially aware clustering. What you see here as well is several other methods that they compared themselves to. So I just wanted to put them out to show that there's many methods more than this method. I think what these kind of methods also allow is to actually define, like, you can find cell types in your data that you wouldn't find without taking spatial information into account. So if the cell types, if the transcriptomic differences are very subtle, then it can help to take the spatial microenvironment into account. And here they highlight that this is a clustering using spatial information into account, and they can identify this MLIs cell type. If you don't do that, if you just do the kind of standard single cell RNA sequencing approach, they were not able to pick up this cluster. This is spatially aware clustering. There's a closely related concept called niche detection, right? Spatially aware clustering is a bit like, what you want to do is you want to identify clusters that are both transcriptionally similar and they are spatially similar. They're kind of close. In niche detection, you want to look for spatial neighborhoods, or so here I think you still want to learn something about, so the difference is here you still want to learn something about the individual cell. In niche detection, I think you want to more learn something about spatial neighborhoods. You zoom out a little bit and ask, like, I mean, you wanted to learn something about the microenvironments themselves. I won't have time to go into the details of how that works, but similarly, what you do is kind of, you don't look at individual cells, but at cells that are close by. I'm using an example called Niche Compass here, which is based on autoencoders, so an AI-based tool. And one of the examples that they show, and that I quite liked, was this, where they look at different mouse embryos. This is like replicates of a mouse embryo. And what they found is or what they could do is, if you just kind of annotate cells in space, you kind of here find forebrain, midbrain, hindbrain lineage of cells within the embryo. If you take the local neighborhood, so the niches, into account, you can get a way more fine-grained result, and you, for instance, can resolve transcriptionally the floor plate, which is marked here in these embryos, which you couldn't do before. So once again, this is something to, in a way, learn something about how cells are arranged in space. In this case, learn something about the microenvironments, where certain microenvironments located. Okay, ligand-receptor interaction. This is something that I think Katrin didn't introduce that, but ligand-receptor interactions is something where the idea is, you can do it, this comes from single-cell RNA sequencing. It's basically, we know, we have some prior knowledge about which cells communicate. So this is about learning cell-cell communication, which cells communicate with other cells via ligand and receptor pairs. And basically the idea is that there are now appearing more and more tools that will look for ligand and receptor pairs and check whether they are spatially correlated. And if they are correlated, this kind of, you can use that to hypothesize that cells are communicating. So there are these ligand-receptor interaction tools. One of the example, if you want to check that out, if this is something, if you're interested in cell-cell communication, is Liana Plus, which integrates a lot of methods that are available. I can recommend that. I worked with that before, just single cell, not for spatial data. And maybe the last point that I want to make is now, how can we integrate spatial data sets with other data sets? And one obvious candidate, I think, is single cell RNA sequencing data set, because often that's available. And I think there's two main things that we can do. One is cell type deconvolution. The idea here is, and I exemplify this with a tool called cell to location, is that when you have a specific tissue and you do two experiments, you do a single cell RNA sequencing experiment and a spatial transcriptomics experiment of the same tissue. Here, mainly think of this now as a Visium, not Visium HD, but a Visium dataset where you have these dots which might contain something like 100 cells or something. And what this cell to location tool does is it asks, or it answers the question: if on this spot I now measure a certain transcriptomic profile, what would be the cell type composition which I can predict from the single cell RNA sequencing dataset? So if I have 3 cell types in my original dataset, for each of these dots, I would get the composition of these 3 cell types. And then I can do that all over space. And that way I can kind of get back some resolution, some single cell resolution into the spatial data by leveraging the single cell RNA sequencing dataset. There's many more cells. I think the hands-on workshop will use RCTD, which is called robust cell type deconvolution. You can use this within the Surat framework. There's DESTVI and Spotlight. I think from these tool and this tool, cell to location, I think there's a benchmark saying that cell to location performs best, but it's computationally the most intense. There's also new approaches coming from the topological data analysis world, a tool called TopAct, and Heather Harrington will actually talk about that tomorrow. One more thing, one last thing that I want to talk about is you can also impute gene expression if 4 missing genes in panel-based assays, right? So in a Xenium assay, we might have only 150 genes, 300 genes, something like that. But there's 30,000 genes altogether. And so the idea there is that you can combine the single cell RNA sequencing data where you have the full transcriptome together with your spatial data to impute genes. One tool that you can use is Tangram. And basically, I think this sketch just exemplifies what I just said without into details. You basically use the two things. I think that the intuition behind that is if you, in the Xenium dataset for 100 genes, you know how they are expressed where in space, then you use these 100 genes to line your single cell RNA sequencing set, right? You have the same cells in the single cell RNA sequencing. And from that, then you impute the genes that you didn't have in space. And I think one beautiful example here is a work by the lab of Alex Shear, which was published on bioRxiv last year. And here they show, they did whole embryo spatial transcriptomics of the zebrafish. And what they show here is in red, these are actually things that they measured by merfish. And then in blue, there's all the genes that they imputed from the single cell RNA sequencing data. Of course, for some of the genes, they tested whether this is working by leaving away some of the merfish genes. And that, I think if you probe that, nicely captures the variability of your cell, these methods work pretty well. All right. I think we're going to share the slides later on. So I have a few links for if you want to get into analysis, into coding yourself, how to get started. There's this single cell best practices book, which also features the chapter on spatial, which I can recommend. And there's some vignettes by the Python world and the R world here. That's all I wanted to share with you. And I'd like to thank you for your attention.